Okay, are there any questions? Can somebody,
Do you mean about the lab, Professor? Or
yeah, do you have something specifically that's not lab?
um well, can it be about the previous lab or just the lab 6?
No, it can be about a previous lab.
Okay. Well, about for lab 5, I had a question about the specificity of the quadrants. The way I understand it, right? When we re-sterilize the loop, isn't that a way of essentially diluting the culture? Is that what it is?
I'm not sure. What's your question? What are you
I'm
asking?
I'm
What's the
ask,
question?
I'm asking whether I'm understanding it correctly.
What's the question? I don't understand what the question is.
Is the loop when it is being re-sterilized for the quadrants, when we put it in a Bunsen burner,
You
is
sterilize the loop before you streak it on a quadrant.
right before each quadrant, right?
Yes, normally.
So so my question is it a way to dilute it, right? I mean, Pasteur
Oh,
was
yeah, yeah,
dilute.
now I understand what you're asking.
Okay.
When you sterilize the loop and streak it, the bacteria, you obviously cool the loop, pick up the bacteria, streak it on quadrant 1, then you sterilize the loop, and
Hmm.
then to streak quadrant 2, because
Hmm.
the loop is sterilized, quadrant 2 will be more dilute than quadrant 1.
Okay.
Okay?
Okay. So you don't. So on the contrary, if we had not done that, we would just drag an excessive amount of it, right, across the quadrants.
This is one of the questions for one of the labs. If you do not sterilize in between the quadrants, notice how the
Yes.
question doesn't say it's not sterilized before quadrant 1, but in between the quadrants, then you're
Hmm.
not diluting the bacteria from quadrant 1 to quadrant 2. So quadrant 2 will have the same bacteria as the end of quadrant 1. It won't be diluted.
Oh, okay.
Okay.
Hmm. Oh, I see. Okay, makes sense. That makes sense. Um, for hemolysis, I had a question about hemolysis.
I'm
Am I
sorry.
from saying it if I'm saying it correctly? Hemolysis or
Hemolysis. Yeah.
hemolysis. Uh, my question is there other benefit other than the iron, right, that you would expose? Was the benefit to the organism, to the pathogenic bacteria? I mean, I understand that they
Okay,
can
what's in a red blood cell? If you're lysing the red blood cell, what's in a red blood cell?
iron,
What else is in a red blood cell?
hemoglobin,
What else is in a red blood cell? Or any cell
protein,
for that matter? Protein. What else?
amino acids.
Everything. Yes. All right. You got it.
Okay.
So, what benefit will a bacteria get from lysing a red blood cell?
Feed and all the nutrients that are in there.
Mm-hmm.
Okay. All right. Sounds good. Thank you.
All right. Let me, oops, get ready with lab 6. Am I showing my screen yet?
Not yet.
Like I'm not. No, okay. All right, let's share my screen. Make sure I'm going. Yep, got the red dot there. Okay. Don't really need that one. All right, so today is April 27th. and we should be doing lab 6. Let me remind you, you need to read the unknown project instructions. I'm going to talk about that next Tuesday. We will start the unknown project. Let me see if I still have that open. I don't think I do. Yeah, I've already closed it. Sorry. You can find the unknown project instructions in the lab for, it's probably going to be week 5. All right, today we're working on lab 6, disc diffusion and antibiotics. Any questions about any of that? If not, boy, that looks like we have a virtual lab today. You know, I imagine we do have a virtual lab today. Let me check here just to make sure that's not an older version. Yep, we do. Okay. So there's some reading to do for lab 6 on disc diffusion, the evaluation of disinfectants and antiseptics and antibiotic effectiveness. Read page 177. There's a little bit to read about the principles of effective disinfection and evaluating a disinfectant, and then the different types, page 187 to 194. Read about chemical methods of microbial control. That's just 2 pages, 196 and 197. Read about antibiotics, antibacterial drugs. That's really 1 page, 554 to 555. Take a look at table 20.3, and then some test guides to chemotherapy, the diffusion method, 2 pages to read there, page 567 to 569. In today's lab, we're going to be having some video clips and a virtual lab. The learning objectives for this lab are: 1, identify the type of a chemical agent and the active ingredient used in various disinfectants and or antiseptics. 2, evaluate the effectiveness of disinfectants and or antiseptics using the disc diffusion method. 3, be able to determine the mode of action of various antibiotics. 4, evaluate the effectiveness of various antibiotics using disc diffusion. And most of the lab will be talking about 4, evaluating the effectiveness of an antibiotic using the disc diffusion method. 5, be able to list the variables that influence antibiotic disc diffusion, meaning the disc diffusion method, which is called the Kirby-Bayer method, or here it's called the disc diffusion susceptibility testing. Most of the time, I'll call that the Kirby-Bayer method after the 2 doctors who discovered it, and be able to define the terms. Know what sterilization is, disinfection, disinfectant, an antiseptic. What's bactericidal, bacteriostatic? What do these 2 terms mean? What is an antibiotic? What's the spread plate technique? And what is a zone of inhibition? So when we're evaluating disinfectants and antiseptics, it's important to know the term. Sterilization refers to or the removal or destruction of all viable organisms, including endospores. When we're talking about we sterilize an object, it may not mean prions, but endospores are removed by sterilization. In the lab, how we get sterilization most of the time is by putting something in an autoclave, which is like a giant pressure cooker. Disinfection refers to the destruction of vegetative cells, not necessarily endospores. A disinfectant is a chemical agent applied to an inanimate object. An antiseptic is a chemical agent applied to living tissues. So the same chemical could occasionally be used as a disinfectant when you're applying it to an inanimate object, Or if the chemical is applied to something that's living, we call it an antiseptic. For example, if I were to clean my toilet with bleach, it would be a disinfectant. If, on the other hand, my cat, which I don't have a cat, were to get I don't know, a cut or something on its paw and to get infected, and I were to treat it with bleach, that would be an antiseptic. Usually antiseptics are not as strong or corrosive as a disinfectant, but bleach occasionally is used as an antiseptic, and obviously it is harmful to the skin, but it used to be used as the primary means for stopping necrotizing fasciitis. You'd wrap a cloth in bleach and then wrap it around your area that had necrotizing fasciitis. And they found that that was a good way to stop necrotizing fasciitis. I don't know if that method is still used, but it was very commonly used. When was that? In the 1990s, in the year 2000. To evaluate the effectiveness of a chemical agent, the surface of a nutrient agar plate is often inoculated with a particular bacterium. And you usually grow the bacterium in a lawn of bacteria, meaning you get the bacteria to grow everywhere. And then when the bacteria have been absorbed by the plate, because you usually put in a liquid culture, so you let it absorb for about 5 minutes, then you put a paper disc saturated with different chemical agents applied to the surface of the agar. The chemical agent then diffuses into the agar, establishing a concentration gradient around the disc. You then incubate the plate, and after overnight incubation, the bacterial growth pattern around the disc is observed. If there's no zone of inhibition, obviously that agent did not stop that bacteria. If there is a zone of inhibition, then the chemical is at least a little bit effective for that particular bacterium. That doesn't mean that if we have an antibiotic in the disc, that doesn't mean that the bacterium is sensitive to that antibiotic. It just means that the bacterium is a little bit sensitive to that antibiotic. And for sensitivity and resistance, we'll discuss that in this lab. All right, this table shows you the different chemical agents, their general action, and their preferred use. You don't really need to memorize this table. I could say for the final, when we're going to talk about the different chemical agents, that probably you should know most of the left column. The general action I won't quiz you on, and I won't quiz you on the preferred use also. But for this lab and all the quizzes, you don't need to know that table. The zone of inhibition size depends only partially upon the sensitivity of the organism to the chemical, like the disinfectant or the antiseptic or the antibiotic. There are various other factors that affect the zone size, including the concentration of the chemical. Generally, if not always, you get a larger zone of inhibition the more concentrated the chemical. The molecular weight of the agent, the larger the molecular weight, the smaller the zone of inhibition. And then the density of the bacterium inoculated on the plate. The higher the bacterial density, the smaller the zone of inhibition. So when evaluating antibiotic effectiveness, we say an antibiotic is bactericidal if the antibiotic kills the bacteria. On the other hand, if the antibiotic only inhibits the bacterial growth, we say it's bacteriostatic. And how you can tell the difference between bactericidal and bacteriostatic is you can remove the antibiotic and then see if the bacteria grow. It will be, if there's growth, it'll be bacteriostatic. If no growth, bactericidal. And then another way of doing it is you can streak the area. And if you get growth, the antibiotic is bacteriostatic. If you get no growth, the antibiotic is bactericidal. When a bacteriostatic antibiotics are used, the immune system is required to assist in the elimination of the infection so that an antibiotic, which is bacteriostatic, does not remove the bacteria from the patient. What you do is you stop the bacteria from growing, and then the patient's immune system removes the bacterial infection. Any question about any of that? Okay. The Kirby-Bauer distaffusion test is the most commonly used method for testing an antibiotic. Excuse me here. I feel like I'm going to sneeze, so I'm blowing my nose to prevent that, but we'll see if it works. So the Kirby-Bauer distaffusion method is a method we use to test antibiotics to see if they work against a given bacteria. The results help a physician choose which antibiotic to use when treating a sick patient. Bacteria are isolated, grown as a pure culture, and then plated onto an agar plate. Let me see if I can blow this up. We usually put the bacteria on the plate so that it grows everywhere. And you see this whitish area because that would be the agar dark area there. The bacteria are growing essentially everywhere, and that we call a lawn of bacteria. Usually plate the bacteria on using a swab and use a liquid culture. You then allow the agar to absorb the liquid for about 5 minutes, and then you put the filter disc on, and they're impregnated with a certain antibiotic. Yeah, let me see if I can blow that up again. I can't really read anything here, so anyone see any things there? I can't read them, but they have names on them, not names, but chemical symbol, like P will stand for penicillin G. I think AM stands for ampicillin, and I'll show you a table of that. K or Ka stands for kanamycin, TE for tetracycline. And they usually have a number on it, and that stands for the concentration of the antibiotic put on the disc. Anyways, you put the antibiotic on the surface of the agar, and then the antibiotic forms a gradient around the disc. So there'll be a concentration of antibiotic around the disc. If we see a zone of inhibition around the disc, that means the antibiotic is at a concentration high enough to at least inhibit the bacterial growth of the bacteria, if not kill the bacteria. So this has a very large zone of inhibition, and the antibiotic obviously is stopping the bacteria from growing. This has a small zone of inhibition, but once again, it is a zone of inhibition. This one has little or no zone of inhibition, and same with that one. Well, that looks like it has a partial zone right there. We're going to ignore that for the moment. We'll talk about that one. Little or no zone of inhibition. And obviously, this antibiotic is not effective on this bacteria. It's hard to say whether this one, this antibiotic is sensitive to this bacteria. This one probably is because that's the largest. However, you have to measure the diameter of the zone of inhibition and then look it up in a table before you can determine if the bacteria is sensitive to this antibiotic. And we'll talk a little bit more about that when I show you the table. So the inhibition zone size is dependent on several factors. The diffusion rate of the antibiotic. The smaller the antibiotic, the more it will diffuse. The lower concentration of agar in the plate, the faster it will diffuse. Getting this confusing now. The degree of sensitivity of the microorganism, the more sensitive it is, the higher the zone of inhibition. The growth rate of the bacterium will also affect the zone of inhibition. So many things can affect the zone of inhibition. We have to standardize the variables when performing the Kirby-Bayer diffusion antibiotic sensitivity test, also called the Kirby-Bayer method. What we do to standardize it is labeled in this table. We inoculate the bacterium in a nutrient broth using several bacteria colonies, and the bacteria have to be growing in the colonies on a non-selective media. The bacteria have to be less than 24 hours old. You then grow the bacteria until the turbidity of the broth is 0.5 McFarland units. This is approximately 1 × 10⁵ cells per mil. You then inoculate the bacteria onto a Mueller-Henton agar plate. This is a specific media. If you use a different media, you may get a different zone of inhibition. The pH of the Mueller-Henton agar plates are between pH 7.2 and 7.4. Apparently, the pH can also affect the zone of inhibition. The plates can't be more than 150 micrometers in size, meaning we always use the same size plates. And you inoculate the plate with a non-toxic swab using the spread plate technique to obtain confluent growth. We'll talk a little bit more about that when you do the lab on this. You then place the disc. Well, you let the liquid and the bacteria sit on the plate for about 5 minutes, and that's to absorb all the liquid. After 5 minutes, you then place the disc on the plate 24 millimeters apart and place no more than 12 discs on the plate. I don't know if there's 12 there, but that's close to it. The point is you don't have two close together and you don't have all of them far apart. Oh, there it is right here. Add antibiotic disc, 5 minutes, but no longer than 15 minutes after you add the inoculation, adding the bacteria to the plate. So you let the bacteria sit on the plate for 5 minutes, and that's to keep the liquid to be absorbed by the agar, and then get the bacteria established for 5 to 15 minutes. But it's not going to grow in this time. And you then apply the plates between 5 minutes and 15 minutes. You then incubate the plates in a non-CO2 incubator for 16 to 18 hours, grown at 35 degrees Celsius. And you put the plates in the incubator no longer than 15 minutes after adding the antibiotic disc. And you use the procedure to standardize the procedure so that regardless of who sets up this experiment, assuming they're using the same bacteria and the same antibiotics, then their zone of inhibitions will be about the same, if not the same, for each person setting up this test wherever they are in the world. if you did not use the standard procedures, then the zone of inhibitions would be different and they'd be all over the place. All right, after incubation, the inhibition zone size are then measured in diameter using millimeters. You then look at the table set up by the National Committee for Clinical Laboratory Studies, and that's the NCCLS table, that sets the standards for interpreting the zone size, whether the bacteria is considered sensitive or resistant to that antibiotic. And you can note that there is an intermediate size, which is in between sensitive and resistant. And let's just go take a look at the table. So for the antibiotic penicillin G, the G isn't mentioned here, for the organism Staphylococcus aureus, the bacteria is resistant to penicillin G if the zone of inhibition is equal to or less than 20 millimeters in size. On the other hand, Staphylococcus aureus is sensitive to penicillin G if the zone of inhibition is greater than or equal to 29 millimeters. And then between that, we say that Staphylococcus aureus is intermediate to penicillin G. Each antibiotic and each organism will have different numbers for what size they are resistant and what size they are sensitive in their zone of inhibition. Any question about any of that? All right, so you should know the terms of this lab. And while you're not going to be physically performing any of these activities, everything's going to be done on lab, online, you will be responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bauer disc diffusion test. We have a little video for you to watch on preparing a spread plate, and then review the steps for using a aseptic technique for preparing a spread plate. Let me go through this procedure here. So we're going to presume I really need something a little bit roundish. Don't have anything roundish, so we'll try this. Can you guys see that? Anyone? Hello? Is anyone there?
Yes.
Okay.
Yes.
See that? You can, okay. All right. So you get your cotton swab. We're going to pretend this pen is a cotton swab. You dip it in your bacteria, and then on the sides of the tube, you get off the worst of the liquid so that it's not dripping and it's not totally confluent. You just get off the worst of the liquid. And then you take your cotton swab and you start streaking it everywhere on the agar plate. And then you rotate it a fourth of a turn, and then rotate the plate. I don't know if you can see that, a fourth of a turn. And then you do it a second time, streaking everywhere. And then you rotate the swab a fourth of a turn, and then rotate the plate a fourth of a turn, and then do it a third time, swabbing everywhere. I don't know if you guys can see that. And that way the bacteria will be growing everywhere on the plate. I once had a student who went, ooh, do, 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 oop, do, 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 oop, do, 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 do. And she had these vast holes of empty areas on her plate. And then she couldn't measure the zone of inhibition because the holes were greater than the streaks. And so you do need to streak everywhere. And then when you're all done streaking 3 times, you take the cotton swab and you take it around the edge of the agar plate. The reason is that when you're streaking, it's kind of hard to make sure you get the edge all the way. So you move along the edge of the agar plate to get the bacteria to grow everywhere on the plate. You then allow the plate to sit for 5 minutes at least, up to 15 minutes, to absorb the liquid and the bacteria. And then you put the disc, paper disc, on the plate. You then incubate the plate agar side up, upside down, in other words. Agar side up, meaning the lid on the bottom, overnight at 35 degrees Celsius. All right, number 3, you don't need to answer. Procedure 2, evaluation of antibiotic effectiveness. View the following video clips and complete the virtual lab. So we have a video on how to prepare a culture for Kirby-Bauer diffusion, and then another video on interpretation of the results for Kirby-Bauer diffusion. We also have a virtual lab for doing the Kirby-Bauer, oh shoot. Now let me leave that there. I thought I updated that one. Up. Let's see if it's on here. No, it's not. Let me get that link. There we go. This may take a little while to download, so give it time to download. You should see this. You can read about the description, the steps you have. We're just going to start right here. And then what you do is you take this ruler and then you measure the zone of inhibition. So for AMC, it looks like it's about 6, no, make that 11 or 12. And so select what it is. This would be resistant, susceptible, and then intermediate. This is 12 or less. So it's susceptible, resistant. If you pick the wrong one, it'll give you an X through it. Okay, but if you pick the right one, it'll be green in color. And then cephalus then is next. This one looks like 30. So greater than 18. Right there. Let's do it wrong first time, and then do it right, green. So that's how you do this. You can click on this and it'll call up another plate and do the same, in which case most of them don't have a zone of inhibition, but that one does. And then click on it here. And there's a third one. And Staphylococcus aureus, which actually looks like only that one has a zone of inhibition. 16 or 17. Vancomycin. Yep. All right. Any questions about any of that? Oh, let me copy that into the one I want. I sworn I did that last term, but I'm not sure. Maybe I didn't save it or something. Then two, use the standards required for the Kirby biosusceptibility testing in E. coli culture using the spread plate technique, meaning the Kirby Bayer method. Antibiotic discs are placed on the surface of the agar and incubated overnight at 35 degrees. And below are the results and the NCCLS zone standards table used for interpretation. So here are the zone of inhibition for ampicillin, cephalopathin, gentamicin, and tetracycline. You need to measure this, and you'll note that for measuring the caliper, the 0 does not begin here. It begins here, and so you have to measure 0 to 0. So this is 15 millimeters, or just a tad less, instead of a tad less than 10 millimeters. Okay, everyone got that? So you're reading the caliper 0 to 0. And then come down to the chart. So I'm going to do the first one for you. You can type right in the chart. Make sure I got the right one. Ampicillin, I'm going to just say 15 millimeters. And the interpretation, 15 millimeters, is right here, is resistant. All right, any questions? So fill in this table. And then 3, list the variables that must be standardized when performing an antibiotic susceptibility test using the Kirby-Bauer diffusion method. And the lab module does tell you the variables. It's in a table. And you need to fill out each, what is that, asterisk? And then answer the questions. 1, the size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? 2, what would happen if we didn't use a septic technique to put the antibiotic disc on the plate? And you should define that. So tell me what happens in the results of the test. Okay. And I need to put that in your copy because you guys don't have that yet. Three, what would happen if we increased or decreased the concentration of the antibiotic in the disc? You must give an answer for both increased and decreased the concentration of the antibiotic in the disc. 4, antibiotics can be bactericidal, that is, they kill microbes. I've lost my mouse, there it is. Or they can be bacteriostatic, they inhibit microbial growth but do not kill the cells. When observing a zone of inhibition around a disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? This is a thinking question, and students often get this one wrong the first time, so you need to think about that one, although I did mention it in today's lab. When performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized, meaning we use the same kind of media, Mueller-Hinton. We have the agar depth, the same depth. I never went through the different standards, but the agar of the plate is always the same depth. And then the age of the culture. We always use cells which are less than 2 days old. I think they're actually less than a day old. And then we use cells that have been growing to 0.5 McFarlane units. And that's just a way of standardizing how many cells there are per mil. And 6, are there any other substances besides antibiotics that we could evaluate using this method? You do need to answer more than yes or no. You have to name the substances. You should probably name 2, but I guess 1 would be fine. Although it does say substances, so that would be 2 or more. Does the presence of the zone of inhibition necessarily mean that the bacterium being tested is susceptible to the antibiotic? Once again, this is a thinking question, but if you understand this lab, this should be a really easy question to answer. All right, any questions about that? Now remember, do not put your answers in the lab manual. Put it in the worksheet. The worksheet is much smaller and it will not fill up my Canvas website. If I ever get too many things, too many long projects, my website will fill up and then you guys won't be able to submit your assignments. All right, I'll be here until 8 o'clock or the last student leaves to answer any questions. If there's no questions, I'm going to say get started on the lab. Come on here. Share my screen and state that I did forget something, and that is when we're doing the Kirby-Bauer method for testing the sensitivity of one strain of bacteria against different antibiotics, the Kirby-Bauer method is actually a very good procedure to test the antibiotic. And the reason is when we give antibiotics to a patient, the antibiotic has to diffuse out of the bloodstream, and go in, diffuse into the tissues to be effective against that bacteria. And so the Kirby-Bauer method, testing the diffusion of the antibiotic out of the disc into the agar plate to make the zone of inhibition, is an actual very good method for testing the sensitivity of the bacteria, meaning it's highly correlated to how we want to use the antibiotic against the bacteria. When we're using the Kirby-Bauer method to test a disinfectant, it's not really that effective because what it's measuring is the disinfectant's ability to diffuse across the agar plate. And when we're using a disinfectant, we're usually applying the disinfectant directly to a surface to kill the bacteria. We're not having the disinfectant diffuse across anything like the agar plate. So for disinfectants, the Kirby-Bauer method is not terribly applicable because we just don't use disinfectants in this way. However, it is very effective in testing antibiotics because we do use antibiotics this way. Any questions about that? All right. I just wanted to state that because I forgot to mention it.